I'm going to mute myself, and we look forward to your talk.
All right, I'll just close my door. Well, thanks, you guys for inviting me to give this seminar. So I'm just going to give you a flavour of what we've been doing here at Stockholm in relation to planktonic forams in the high Arctic. So there's not that many sort of institutions that have access to a lot of material from the really high Arctic. So coming to Stockholm has been a real treat. So I've been working sort of all over the Cenozoic over the years. And when I started in Stockholm and the opportunities to start looking at the Arctic came along, well, I thought, how difficult can that really be? There's only one species, right? Pachyderma. Isn't that a bit boring? Well, I've been extremely wrong about that. There's all sorts of really interesting problems, questions, and that's given me a lot to occupy myself with. So this is just some of the things I hope to cover today. So I will start with a bit of something on the sort of Swedish Arctic coring legacy. Say something about the environment itself, the Arctic Ocean and the environment, and what we know about planktonic forams in the Arctic. Then we'll have a look at the past, the Arctic sediment pile in the Arctic, and where and when we have forams there. This will include a look at some interglacials, which have become, or what we think are interglacials. We might not know exactly which one at this point. And for that, I will talk about the genus Turborotolita. And something that's really has been unfolding the last 5 or so years for me is some explorations with modern planktonic forams. So an opportunity to sample wild planktonic forams in the Arctic. All right. So here's a really wonderful bathymetric map of the Arctic Ocean, and we were very honoured to have Martin Jakobsen, who was just fixing the PowerPoint presentation actually for us. So it was Martin and Jan Backman, first of all, that kind of started my connection with Stockholm after I met Jan on the Georgish Resolution, I have to say. And it turns out there's a real history of working in the Arctic with the Swedish icebreakers at its center. So going back to the 80s, there was the Irma icebreaker, and later on from 91, icebreaker Oden, and the Swedish Polar Secretariat have been very much behind that adventure, both sort of their duties breaking ice, but then also they've been hired into the research arena. So over the years, there have been many Oden expeditions. I think there's now 80 or so cores. Martin Jakobsen's a real expert on bathymetric surveying and geophysics in the Arctic. And then there's a whole treasure trove of Arctic cores sitting in our basement, which is super exciting. So initially, having worked with the Eocene a lot and the Eocene-Ligocene transition, it took me a while to sort of get into the Arctic, but now I realize there's a myriad of questions, both sort of past and present, when it comes to trying to figure out what we've actually got in the Arctic, what species are there, what's the fossil record, what's the signature for interglacials, how can that be used for paleoceanography? But then what do we actually know about the planktonic forams in the modern? And in terms of also understanding how we can better use them as traces when we do find them, and are we finding signs of change as things like Arctic Atlantification proceed? So thinking then about the Arctic environment as a pelagic environment. So here's a nice oceanographic circulation map with a bit of 3D here showing some of the important upper ocean water masses. so that our planktonic forams are going to be sort of living and interacting with. So really important for the Arctic is that it's this bizarre, very internal land-bordered ocean basin. There's ridges running across the middle of the basin, and it's got these very narrow gateways, the Fram Strait coming from the Atlantic and the Bering Strait, which is only very shallow on the other side. So really important for the ocean circulation is the inflow of Atlantic water through the Atlantic Gateway in the Nordic seas. But really importantly, that Atlantic water, which is warm, relatively warm and salty, it sinks below the polar layers north of Svalbard. So if we look at a cross section of this, we can see that this Atlantic water sort of sits in the upper sort of 100 to 400 meters in the Arctic, and then it's circulating around the basin margins, perhaps doing a few turns in the middle, and then you have this much fresher, colder polar surface layer on top. So our forams are sort of surviving in this environment. The high Arctic is still today characterized by thick perennial sea ice cover. In terms of the planktonic foram environment, so here's a sort of very idealized schematic of their environment. So what we've got here is our sort of water masses. Here's the Atlantic water now that's come in via the Nordic seas gateways, and then it sinks below this thick Arctic halocline about north of Svalbard, and then it sits there and circulates. So it's a kind of a strange ocean in this respect, that in the warmest water mass is in the middle or deepish. In terms of planktonic forams, in the sort of subpolar realm, in the Fram Strait, Barents Shelf, we've got pachyderma, and in some parts we've got a lot of Turborotolita quinqueloba. Very quickly we lose quinqueloba, and then we are in the sole domain of pachyderma, the true polar species. Okay. All right. So what about forams, planktonic forams, carbonate back in time in the central Arctic Ocean? Well, we don't have very much knowledge on that. The only long-term hole, oh, my dot has moved, it should be there, is the single IODP expedition that has managed to get into the Central Arctic, the ASEX expedition from 2004. That expedition was successful in drilling about 400 meters on top of Lomonosov Ridge, and what was discovered there was really pretty revolutionary at the time. Just to simplify, we've got this this unit 2 was basically sort of black shales, and above that you transition into this sort of silty clays. Most of it is very, very microfossil poor, or carbonate microfossil poor. Although the bottom part, the Middle Eocene and the PETM below, contains silicious microfossils and diatoms. The upper part, we've got ice rafted debris appearing what appears to be sometime in the Middle Eocene. Planktonic foram barren for much of this, but we do, from some part that's the coarse age model suggest is about Miocene. From Miocene time, you move into a agglutinated benthic facies. So that agglutinated benthic facies appears to continue for, well, it's there. The transition between calcareous and agglutes is about 2 to 6 meters subsurface. So of this 400 meters, most of it is carbonate carbonate free, which is a little bit sad. But what is there above this level, and there's kind of grain size indicators of increased evidence of northern hemisphere glaciation at around 53 meters. And in the top kind of 8 to 10 meters, there's a characteristic sequence of light, dark cycles, sediments, with the dark sediment layers appearing to be glacials, and in those you get interglacials, and in those you get pachyderma. So that's kind of the nutshell, mostly of the long-term history of planktonic forams in the Arctic. I would just like to say that the age models are pretty tricky. The paradigm is that there's a massive hiatus, 28 million year long hiatus separating the Eocene from the Miocene. There is alternate age models to that by Hilaire Marcel, and people are still debating this. Now, in terms of when did pachyderma arrive in the Arctic, the paradigm based on biostratigraphy calibrated to paleomagnetics outside of the Arctic, so in the Nordic seas and North Atlantic, is that typical encrusted pachyderma, probably with a polar specialization, evolved about 1.8, 1.7 million years ago. So it's possible that arrival of pachyderma here can be sort of dated to that time interval. The overriding problem is that chronostratigraphy in the Central Arctic is super challenging. PMAG is unreliable, radiocarbon is confusing, biostratigraphy is limited due to both the low diversity and preservation challenges. But I think there's hope. This is an active area of research, including especially the nanofossils. There's a bit of a resurrection in nanofossil work on this. Anyway, let's have a look at a microscope view of high Arctic planktonic forams, and you can see where this sample comes from. This would have been one of these well, this is actually from a core top, so 0 to 2 centimeters, and this is very typical for a foram-rich layer, and this is right in the middle of the high Arctic there. Basically dominated by pachyderma, mixed in with a lot of quartz ice rafted sand. You've got the odd pteropod in there. If we zoom in, this is another sample. Again, lots of nice pachyderma of a variety of different morphotypes, I have to say. When I first started looking at the Arctic, I had to get my head around this diversity of different pachyderma morphotypes. And that's something that we're returning to more and more. You get the odd sponge spicule, various benthics. So it's telling us that there's a healthy ecosystem with food and various different organisms participating in that pelagic ecosystem. So just going back to connecting this with the timing of pachyderma arrival, if we go into the Nordic seas, and I've just had explorations of some of the old studies on this. Here we've got in blue. This is work from the Voring Plateau by Spiegler and Janssen back in 1989. What you see go back in time is where you have carbonate is a sudden appearance of pachyderma. And there they do have magnetics. So this has been calibrated against the paleomagnetic reversal stratigraphy. And this is where you get this number of about 1.7 million years ago. Before that, you have a sort of Morbilloides, a whole selection of other Neoglobocadrina species, including Atlantica, which is a real important species in the Miocene to Pliocene in the Arctic. They disappear, and you get this encrusted pachyderma species. Going a bit further up into the Arctic, into the Fram Strait, Spiegler did something similar, but here you've definitely got less carbonate and fewer planktonic forms generally. But again, they've been able to calibrate this arrival of, so where the blue then is pachyderma occurrence, and it seems to arrive fairly abruptly in quite a few sites, corresponding to the onset of the Quaternary really, and strengthening of northern hemisphere glaciation. So it really sort of supports this idea that this type of pachyderma became adapted to this cold polar oceans. So after that work, following this up and following up some of the calibrations by Clement and Weaver in 1986, Huber et al. 1999 looked more carefully into pachyderma size. So also working with the Voring plateau cores, they found that if you look carefully, planktonic forams, and this is mostly pachyderma, start appearing about 1.7 million years ago in the Nordic seas, but then they sort of start getting bigger in line with increased carbonate contact generally in the cores, and disappearance of any other kind of subpolar species. So they put this together to suggest that the increasing size in pachyderma, which you start to see from about 1.1 million years ago, is a real sort of adaptation to the increasing polar conditions, and that this adaptation itself was actually important for carbonate production and carbonate sedimentation in the Nordic seas and the Arctic, and helped us as paleoceanographers, because then we actually had something to work with. So a hypothesis then, before pachyderma adaptation, carbonate might have been sparse in the Nordic seas not because of a few planktonic forams, just because there weren't enough to really help buffer the sedimentation and help preservation. So just going back to our long-term story in the Arctic, then it's possible that this first appearance of the pachyderma we see, maybe that's this sort of 1.1 million years ago special adaptation. That's my new speculation, by the way. Okay, so moving on, I'd like to say something about subpolar planktonic forams in the Arctic. In the core tops we see in the central Arctic, there is no turborotolita. But if you look in a whole bunch of cores from across the Arctic, and these physical properties curves here represent different cores, we find sort of spikes in turborotolita at several horizons in what has been interpreted as the late Quaternary. So here we've got the MIS stages, the marine isotope stages, labeled here. I would just like to say that this age model is rather out of date now. What we think, if we think this might have been MIS 5 up here, we now think that only part of it, or the top part, is marine isotope stage 5, and everything else is older, such that this bottom horizon could be marine isotope stage 17 or 21 or something older. And this is the result of new nanofossil calibrations from Mohammed Razamajui, which is quite exciting. Anyway, age models aside, what is important and interesting is that when I started looking at this and trying to make sense of the taxonomy, we see that the upper morphotypes are this typical species that we recognize, Turborotolita quinqueloba. But the bottom morphotype, the deeper, older morphotype, is a rather odd, different looking thing, which Tom Cronin has called Turborotolita egelida. So just compared to quinqueloba, for quinqueloba fans out there, we know that quinqueloba is one of its real characteristic features is this sort of overhanging teardrop final chamber. These are Turborotolita quinquelobas with that teardrop final chamber from the Central Arctic. I have to say they're very small. These are from the Nordic seas, almost to scale. They're much bigger in the Nordic seas. And then this beast, T. egelida, is quite different looking and quite large, really, and has this very open umbilicus. They're all spinos. You can look under the SEM and they have evidence of spinoholes or spines. So this is really intriguing, this morphotype egelida, and I've spent quite a lot of time trying to sort out its taxonomy. If anybody has any idea of what it could be other than a Turborotolita, I'm all ears. But just focusing then on the upper horizon, my postdoc, now a research fellow, Flor Vermasson, has really followed up this Turborotolita story in the Central Arctic Ocean, which turns out to be super interesting. So just bearing in mind that today in the Central Arctic, it's all pachyderma, to find lots of quinqueloba is anomalous. And so he has found and counted the ratio of quinqueloba and pachyderma in these upper horizons of multiple cores, found this sort of lower horizon, 2 pulses, as had been suggested before, of quinqueloba. And the interpretation is that this sort of subpolar realm fascies has basically migrated into the Central Arctic Ocean during a previous interglacial, which is super, well, that would have been a very different looking Arctic. So using this reconstruction, he's mapped out where you have, so this is the modern, and these are just the sites that we have, these different circles, and this is the modern sea ice extent, so sea ice minimum and then the winter maximum. The reconstruction based on our Turborotolita quinqueloba perspectives, if you use that model for a subpolar environment, is that a subpolar or sort of a Fram Strait like environment migrated into the whole Central Arctic, such that we were left with only a small amount of residual sea ice, which would have been a very different Arctic. So the conclusion here was that this could be some of the first really convincing evidence for massively reduced sea ice during the last interglacial. This is something important for comparing to model simulations and trying to understand the future. It's consistent with other indicators of large-scale Arctic change during the last interglacial, especially some terrestrial perspectives from raised beaches, mollusk faunas, for example, and coastal forests at the Arctic edge, and even some ice core evidence. It's inconsistent with other types of geochemical evidence, so it's the IP25 proxy in the Central Arctic of the Siberian Shelf, and also in the Nordic seas. There doesn't seem to be much evidence for warmth. So these are all things that have to be reconciled. It's possible that you actually get a lot of sea ice exported through the Nordic seas at times. So that's something we're trying to figure out. The sort of mechanism for this was Flor attributed to basically Atlantification, so a shoaling and penetration of the Atlantic layer. into the Arctic Ocean that it started to interfere with the Arctic sea ice. Oh, we got that. Okay, so going back to, so that was the kind of the last interglacial story. This lower morphotype, this thing called Egelida, is another story, not completed story. When we look down the microscope, these horizons with Egelida are kind of amazing. There is basically no pachyderma, which is miraculous. It's just full of these beautiful Egelida type things. It's also got a load of Cassidulina terrettis. Anne Jennings is going to love that. Very happy, lots to eat. Even in the small fraction, so this is 63 to 125, you see these little things. They've actually got an enormous proloculus, I noticed. So what that means is something that we're trying to digest. And we can go to the other side of the Artesian basin and see basically the same thing. Also, tons of terrettis and other types of benthic forams, and implying lots of food. And again, there's hardly any pachyderma there. There's actually some mutant pachydermas. This is some kind of a weird pachyderma. So basically, a very different Arctic during this horizon. Now, this led me into a long story, which I'm going to summarize very briefly here, about what is this thing. So it turns out that Yvonne Herman in 1974, in her studies of the T3 cores on North Wind Ridge, had already identified this. So she'd identified these. These upper horizon, and then that lower horizon, and this is this horizon full of these things. She called it, though, ex umbilicata. She called it globid. All right. Ex umbilicata, Globidrina ex umbilicata. So, and she did which seemed like a perfectly decent species to me. So my question was, what happened to the name Globidrina ex umbilicata? So it's a long and complicated story, but the conclusion is that Egelida turned out to be a name that was used for North Atlantic plankton that Cepheli had named, and he thought it was this ex umbilicata. I'm trying to argue that that's not the case, that Cepheli's species is actually it's a sort of a juvenile form from the plankton, but I'll spare that long story now. What is important is the stratigraphic and paleoceanographic significance of this species, which I'm now going to call ex umbilicata, because I don't think Egelida is actually the appropriate name for it. It's a really distinctive species. It's got distinctive its spinos. You can see that in its wall texture. The absence of pachyderma from the horizon that it occurs at means that something very different. But so I'm trying to figure out if this species can be found outside of the Arctic, which maybe that will help us figure out what age it is, which interglacial it is. It certainly doesn't seem to be marine isotope stage 11, certainly from the Nordic Seas perspective. And I've been talking to Henning Bausch about this. We're going to try and explore that a bit more. That work is still ongoing, but it's going to be a really interesting story. But in the meantime, there's another complication there. If you're trying to look for this ex umbilicata thing, you have to remember and recognize that you could mix it up with a morphotype of pachyderma. So you've got to be super careful. So Anybody who has spent a lot of time with pachyderma will eventually have recognized these different morphotypes, which have been named by A. Nord and then later on explored by L. Barney Atuna. So it's this morphotype that has been called MPS 5, which looks surprisingly and confusingly like this X umbilicata thing, if you don't look at it under the SEM, because it looks kind of light, it's lightly calcified, it's got little bumps on it, but they're not spine bases, but it can really, and it's kind of got quite an open or relatively open aperture. So you've got to be super careful. So just to illustrate some of these differences, this is some of these MPS 5 morphotypes. They've got bumps. They can look shiny, just like this X umbilicata, but they are not spine holes. Their little bumps are little pustules, not spine holes, and you see that on all of anything that you might call this MPS 5. And just to contrast, here's a more fully encrusted pachyderma, and you see intermediates where you see these little pustules sort of coalescing to almost start building this pachyderma ridge. In contrast, X umbilicata has got spine bases, the remnants of either holes or broken spines, and that looks quite different under the SEM. So various people have already made this mistake, including Aksu in 1985, and Herman as well did this in one of her papers. So this is just a sort of word of caution for going anywhere with this X umbilicata thing. All right, and the final part of my talk here, I just want to report on work we've been doing with plankton actually living in the Arctic under the perennial sea ice. So this is made possible by a recent Odin expedition, which was the Synoptic Arctic Survey with SAS Odin 2021, on which I had a work package. So this was actually a biology expedition. It was the design of the expedition was to get a snapshot of the under the ice ecosystem before it's too late, basically. So everything from fish to bacteria, and they hadn't thought of planktonic forams, but I managed to squeeze on a little planktonic foram project. So with the help of my shipboard team, so Flora Massan and Claire Bird, and on shore base we have Cape Darling and my PhD student at Whitecamp, and also we've dragged in Alison Sang into the team. We used bongo nets, multi-nets, and convinced the biologists to drop some box cores as well. So we've got a whole series of plankton nets and paired box cores in, and went right to the North Pole. So we've got North Pole forams, which we're quite proud of. Now, luckily or conveniently or sadly, the ice opened up towards the end of the expedition north of Greenland. Normally it's super difficult to get into this area north of Greenland because this is the sort of last ice area, as they call it, and all the sea ice gets blown by the prevailing winds. So it's super hostile and very, very challenging to navigate there. But they were even able to get some nets there. So I think what this has given us is kind of a unique, super high Arctic planktonic foram diversity perspectives. What it's like, life under perennial sea ice, some perspectives on foram diet. One of the questions was, are there any subpolar invaders? All of this is useful for figuring out our proxies. So data, just very, very new data, I have to say. This is a paper that's just about to be submitted by Floor. So the first live planktonic forams under the perennial sea ice of the North Pole and northern Greenland. So we've got lots of different stations. This is pachyderma abundance compared to all the different physical and chemical properties. And just to emphasize, we've got one station which is a kind of sea ice edge. The rest are perennial sea ice regions, and what we see there is that the pachyderma is living deeper than under the sea ice. So under the sea ice, the pachyderma habitat seems to be a little bit shallower. So under the sea ice, they sort of dominate between 50 to 100 meters, whereas in the sea ice edge region on the Yermak Plateau, they seem to live over deeper depth from the upper 50 to about 200 meters. So that's actually something that has been seen previously by Bay and Carstens and Wafer and Volkmann, and also Matteo Greco and Kuchera's group. They've sort of explored other areas of the Arctic, not the perennial sea ice areas, but I think they see slightly similar things. So what we also found is there is no planktonic or subpolar invaders yet into the high Arctic. There were a few quinquelobers on the Yermak Plateau, so just north of Svalbard, but that's to be expected, and so pachyderma still rules in the central Arctic. Also, what Floor has, it's really super interesting, is using automated machine learning approaches. He's imaged and measured all the plankton, all the pachyderma shells from the different water depths to see if we can see any sort of depth-related signal, life history signal, and perspectives onto genetic migration. And certainly there seems to be an increase in test size with depth. We're still sort of processing that. Something they also saw is differences in cytoplasm color in pachyderma. There seems to be the red ones, seem to be more of them in the upper water column, and they sort of turn green deeper down. So that's going to be in Floor's paper. And then an even newer perspective, so we're really near the end now, is not of this study, but of my talk, is the work by my PhD student, Teerja Weitkamp. So she's sort of also taken up the gauntlet of looking at pachyderma in the nets and taking on the challenge of trying to identify the different morphotypes in the nets, which is not something that's been done before. Typically, that's been sort of the focus of sediment work. So she's made these nice little cartoons of the different morphotypes, and we've got the first data on the distribution of these morphotypes, their abundance at the different water depths. So these are all the different multi-net levels. And then we've also got the sediment. So this is a super interesting picture of differences in test size and morphotypes. She's also going to be integrating size as well in the central Arctic Ocean. So I don't want to blow Teerja's story. She's going to be given a poster at OTU. Hello. So please go and bug Teerja if you're going to be in Vienna. But we're certainly seeing some interesting action in relation to this MPS 5, the lightly calcified form, which perhaps not surprisingly is likely a sort of a younger juvenile stage. Perhaps it's not calcified yet, and we see more of them in the upper water column. Something else is also interesting is the abundance of dextrally coiled specimens. Now there's an interesting story there. Typically, we would expect 3% of a pachyderma population is permitted to be dextrally coiling, and vice versa for Incompta. But we're seeing something a little bit different in the Arctic. There's more dextrally coiling forms. Initially, we thought that this might be wrongly identified quinkballoba, but we now know that's not the case. And we see a difference in the water column compared with the sediment as well. So that's interesting. And Tirja's got some and our team, which involves our plankton, living plankton experts, Claire Bird and Kate Darling, are helping us interpret those data. So again, Alison Hazang is helping us sort of measure these pachydermas automatically. What I think I can just say is what's very interesting about this Arctic environment, it is a bit of a natural laboratory because it's only one species and only one genetic species. I think it's a Pachyderma type 1A, I think. It's only one species, so we can't really mix juveniles or other things up. We've got this natural system, so that's really interesting. So we can basically measure everything more or less, we think, and assume it's this one species, and look at the distribution of size and morphotypes, etc. So we've got large populations, automated approaches to this measuring, and Tirja's looking at sort of coiling, incrustation, and size. Okay, so that's this coiling stuff. So I think I'm just about at the end of what I wanted to say here. Go and see Tirja's poster in EGU. Flora's also going to be in EGU. This is just a sort of taste of what's going on in terms of Arctic planktonic foram research at Stockholm. It certainly got me interested. So the Arctic is obviously currently being massively transformed. And I can see more and more that even if it's just pachyderma or finding any anomalous occurrences of subpolar species, there's a huge amount we can learn from Central Arctic records. We just sort of started to tap into this, sort of standing on lots of shoulders, of course. Once we sort of bring our knowledge together, there's loads of great opportunities to try and learn what happened to the Greenland ice sheet, sea ice, ecosystems under global warming, or what did happen when the Arctic warmed and what might happen in the future. I think the Planktonic forums assemblages holds a valuable position in informing us on this. Just starting with what happened with previous interglacials, how bad will Arctic Atlantification get? Another question is going to be, what about Arctic Pacification? That's a whole nother water mass that's sort of increasing its reach into the Arctic. What does that do to the planktonic foram fauna? Do you start getting Pacific inflows? Our starting challenge or one of the starting challenges is to get the age models in better shape. And that's certainly work that is ongoing. And so amazing effort going on, I would say, with pachyderma at the moment. Julie Mellans and Kasia Zamelchek and others working with cultures and plankton nets from Fram Strait to the Nordic seas are fantastic. And so adding in these wilds population reviews is super useful and help us just use the records and use the proxies better. Okay, I think I'm done.